Very exciting times. I think not just for biostatistics, but for everybody in the life sciences industry, for the adoption of large language models, artificial
Mhm.
intelligence, you name it. Data visualization is another big one. But even before I will speak about those in a moment, but even before I speak about that, there's actually a fundamental shift that's going on right now in the world of biostatistics and specifically within the world of statistical programming. And those two functions are usually tied together. So when I say biostatistics, please know that I mean biostats and programming. And that fundamental shift is up until this point, the dominant software, the dominant programming language for all clinical trial programming of data has been SAS. It's the type of transport files that the FDA accepts into their system for review of clinical trial data. And it's just the legacy architecture that a lot of large pharma, large device, and large CRO have built their data architecture around. And SAS is a great programming language. It's a great tool that does exactly what you need it to do for clinical trial programming. The issue with SAS is that it's closed, meaning that there really are the only improvements or the only additions that can be made to SAS are from the company SAS themselves. And they're doing so, don't get me wrong, like there are improvements to be made, but and they're doing it, but it's pretty standard with what you see is what you get. And this fundamental shift that we're seeing is really towards open source programming languages, specifically around R and Python, R being the main one.
All right, Robert. Well, thank you so much for joining me on my podcast today. To kick it off, can you share your journey and what inspired you to pursue biostats, particularly in the context of clinical research and the whole FDA approval process?
Sure thing, Naomi. First, thank you very much for having me. It's a great honor to be part of your podcast. I think what you're doing here in terms of sharing knowledge, sharing information about the clinical trial space is greatly needed. And there's not a lot of that out there. You see, there's so many podcasts out there, but none of them are really focused on the world that we live in, which is clinical trials. So I applaud you and greatly appreciate you
Thank you.
podcast and having me. So yes, thank you. Okay, your question about who I am and my journey into biostatistics and specifically within clinical trials and how that all relates to the FDA. So, Robert Rashford, I am a biostatistician by training with a master's degree in biostatistics, but I actually didn't initially go to school to become a biostatistician. I went to school to become a teacher in math. So, a math teacher at the high school level. Did all of it, went to student teaching, loved it. It was a great, probably the greatest 6 months of my college career, honestly. It was
Mm.
such a good time. Making an impact, really helping students understand mathematics, which is, a pretty challenging concept. The issue that I ran into, though, was I went to a very small private school and I got my first student loan bill my senior year, and I kind of panicked. I kind of recognized, like, oh, there's really no way to have a home and a family and be able to pay off these student loans for this private education I just paid for. So I audibled very quickly, did a quick Google search of what job will be needed, and literally this was my job search. This was 2012, and so I Google searched what job will be in the most demand, will be the most needed for someone with a math degree in the year 2020. Randomly picked a year. Who knew it would be such a pivotal year?
Yeah.
All the lists, every link that I clicked, all of the sources I checked said biostatistician will be the most
Mm.
serviced job out there. And it turned out to be true. Like there in 2020, there really are not a lot of biostatisticians out there. And still to this day, there really aren't. So I decided to pursue that career, got my master's degree in biostatistics from Washington University in St. Louis, and immediately started working after that at a very small startup contract research organization. And at the time, I was really doing a lot for that company, not just statistics, but I was doing statistical programming. I was doing some data management and database development and medical writing. Really, I felt like that was such a curse. Like I didn't have a work life balance. I was just doing all this work. all the time. But looking back on it, what a blessing. I am forever grateful and forever thankful to that company because they allowed me to jumpstart my career and really learn about the entire journey, the entire data flow that clinical data has to take from entry at the site into the database system, into programming and reorganizing the data, cleaning the data, and then into statistics to analyze it, then into medical writing and regulatory to report it. So from there, I was able to take those skill sets, join a sponsor company to see what interaction with the FDA was actually like. And then I joined a larger CRO before eventually landing at where I currently am. We are a mid-size RCO called ProPharma, where I currently serve as the vice president of biostatistics and statistical programming. And we service clients of any size and within any therapeutic area. And we even do device trials as well. And we have a global biostatistics team and programming team. Very quickly to wrap up kind of where I am in my career now and in my journey to get to where I am, I just recently released my own website called Better Biostatistics. That is a hub for free material, free resources that I've kind of put together over the course of my own journey into the life sciences field that will hopefully help people start and grow their careers either in biostatistics or statistical programming to start, but then hopefully I can expand that into data management, medical writing, all the other various fields, clinical operations that exist within the life sciences sector. So kind of coming full circle back to education. That's my whole journey. That's my whole goal. And that's why I really appreciate what you're doing in terms of helping others learn about what we do. So hopefully that wasn't too long-winded.
No, I love what you're doing, and I appreciate what you said about your journey. So just for kind of knowledge, I also went into sort of a small or mid-size company, and I was wearing many hats. And I think that's one of the things where it's like if you really want to grow in your career quickly, get into a position that's like that, because you get exposure to so many different things and it allows you to really understand the industry more holistically. Like, how do all these different departments, individuals collaborate together to get the final outcome, which, would be like a submission to the FDA for a trial or like a phase 3 results, etc. So yeah, I love your journey and I love what you're doing. It's important to be able to communicate, what we've learned in the industry to those that are new and just interested in getting involved and maybe don't even know, what really goes on in the clinical research field. So I love
Yes. Yes. It can be such a black box, right? Where you
it.
just know that clinical trials exist, but man, are they a complicated song and dance that you have to go through. And so I completely agree with you. The best career advice we could offer anybody who's looking to start their career in the life sciences industry is to be able to experience all of it. Try and find either a smaller company or a role that enables you to not necessarily do everything, but at least be present and be able to understand the entirety of whatever your company or whatever your organization does within the life sciences sector. Because the more you can see, the more you can then evaluate and say, that's the area I want to dive deeper into. It's like when you first enroll in college, right? They make you take a bunch of gen eds so you can kind of get a lay of the land and see what's out there and then help you make some type of determination of, okay, I really like history. I should go pursue a degree in history, or I really like math. I should pursue a degree in mathematics. So I think your career advice is spot on. Absolutely.
Yeah, exactly. It sort of goes, into like, well, what is it that you want to do as a career? Sometimes you need to just volunteer in a bunch of different places, get internship experience, and then
Yes.
it's sort of like that same type of advice. So, what would you consider to be the most significant sort of challenges that you face as a biostatistician?
That's a great question. The most significant challenges, I think there are many. There are many. I think the one that comes to mind the most is, speaking of a proverbial black box, biostatisticians and biostatistics as a field are most of the time treated that way, where you have all of these other functions within the clinical trial. You have data management, medical writing, regulatory, clinical operations. They all kind of just, it's very clear what they do, right? Like clinical operations, I'm starting the site up, I'm collecting the data, I'm cleaning the data at the site, I'm operating on the clinical trial. Data management, I'm managing the data. biostatistics, for whatever reason, is treated like this black box where I don't know what you guys do. I don't know how you do it. Just get it done. Just give me the p-value, give me the tables and listings, and we'll go to the FDA with the results. And that's such a challenge because, and this is going to sound biased, which is ironic because the job of the biostatistician is to eliminate bias from a clinical trial, but it's going to sound biased. At the end of the day, the FDA or any regulatory body really only speaks one language, and that's the language of statistics. They need to see the data. They need to see the results of the clinical trial in order to make informed decisions about, should we approve this new drug? Should this new device be approved or rejected? And so keeping the biostatisticians in the field of biostatistics off in a corner and say, hey, just give me the p-value, I hope this works out, is the worst thing you can do. And it's such a challenge for the biostatistician to raise their hand and say, I'm here, please loop me in on these discussions about how the study should be designed, let alone how the study should be analyzed. So the sooner you get your biostatistician involved in the clinical program design aspect, the greater your probability of having a meaningful clinical trial that'll actually be presentable to the FDA really goes up. Notice that I didn't say the probability of having a successful trial goes up, because clinical trial success is dependent upon so many factors, let alone random chance, but also just, does the drug work? So it's not necessarily a direct, hey, include your biostatistician, increase your chances of clinical trial success. No. What I'm really saying is include your biostatistician earlier to set yourself up to have the best chance of having a meaningful clinical trial that the FDA or any regulatory authority will take a look at. So I would say the biggest challenge that biostatisticians face today is having their voices heard, and having their voices heard earlier in the development process. I think a lot of heartache and a lot of dollars could be saved if companies knew to loop in the biostatistician at the beginning instead of just the end when they typically think of the biostatistician.
Yeah. So, when we're talking about designing clinical trials, what approach do biostatisticians take when they're involved in that process?
That's a great question. And it's very dependent upon the type of study that's being designed. So if you are designing a dose escalation study, for example, an early phase 1 trial, where you don't really know what level, what dose amount to give a subject. So you're going to design a phase 1 dose escalation study. Basically, you'll enroll a handful of subjects, give them a low dose, see if they have any toxic events. If they don't, okay, move on to the next cohort. That type of design, very simple for statistics, not a lot of craziness going on there. You kind of know what you have to do. So at that point, the statistician isn't super heavily involved. Maybe you have a unique dose escalation cohort design, like a Boeing design has become very popular these days. But outside of that, you don't need to calculate a sample size because you're doing these smaller cohorts. You're not really testing for an efficacy endpoint, so you don't need any crazy modeling. So the statistician designing a phase 1 study, minimally involved, you still want them there, but they're not going to be doing anything crazy. As you move into the phase 2 and then ultimately the phase 3, then it's a very heavy give and take type interaction where the drug manufacturer, the sponsor, will say, hey, this is our objective. You always have to know what the target product profile is, the TPP, what that is for that drug or for that biologic or for that device. And the biostatistician will then take that information. Okay, company A, I know that you want to be able to make these claims about your drug. It lowers blood pressure or whatever the case may be. And then the biostatistician takes that information and they'll look at the FDA's history. They should go look and see how has the FDA treated drugs like this before? Have they approved anything in this therapeutic area? If so, what endpoints were used? And so really what the statistician is trying to do is they're trying to answer the question of, in order to make these claims that the company wants to be able to make about their new drug, what measures or what analyses does the FDA need to or want to see? And so it's reviewing what the FDA has done in the past. It's reviewing what guidelines the FDA has released about that therapeutic area. Maybe they have a specific set of parameters that they require to see in a submission. And then from there, once you know what the company wants to do, and once you know what the FDA needs to see in order to do that, in order to enable the sponsor to do that, then the real fun begins, which is, okay, I know what measures the FDA is looking for. Let me design some type of analyses that will enable the statistical measures to generate a p-value or a confidence interval or some type of result that gives the FDA confidence, yes, this is a true signal, this is a true effect that the treatment is having on this disease state. And the statistician is trying to come up with those analyses methods, those analytical methods, in such a way that the sample size of the clinical trial is not super high.
Mm-hmm.
So, not to get too technical, and I apologize if I am, but basically what a clinical trial is it's a tool. It's a way to say, I can't give this drug to everybody and prove that the drug works for everybody. That's just not safe and it's not practical.
Mm-hmm.
So instead, what we do is we give the drug to a very small set of people, and then we look and see how the drug did on that very small set of people. And we use statistics to say, okay, if this drug worked on 1,000 people, if it worked to this degree, to this statistical level, then we have faith, we have confidence that the drug will work for mostly everybody within the population that the drug is meant to work on. So statistics is really trying to be able to say, I've given the drug to this small group, and now I have the ability to say it's going to probably work across the board here. Long-winded, but hopefully that answered your question.
No, it does, and it makes me think about like patient recruitment, and there's guidance that I think came out a couple years ago regarding increasing diversity in clinical trials, right? And so
Yes.
how do biostatisticians play a role in identifying, ways to increase diversity in the trials?
We're trying to do a little bit better of a job at this because it has not been a focus in the industry, at least not to my knowledge, until that guidance really came out about diversity and inclusion. And so what the biostatistician is able to do while we are determining the analysis measures to use to prove to the FDA, hey, this drug works, it's effective, and it's safe, we're doing the sample size calculation to see how small of a sample we need or how big of a sample we need. And you can throw in some assumptions in there. Maybe the drug is going to work differently for an age range of 65 plus versus a younger age range of 18 to 25. And so based on that, you can start to stratify your analysis for different covariates and really start to say, if I enroll this specific race or this specific ethnicity or this specific age range, how will that affect my analysis? And so the statistician is working very closely, and this is a critical point here. It's a very collaborative nature. The statistician it's a very collaborative process. The statistician is working very closely with key opinion leaders in that space. Say it's an oncology drug. They're working very closely with oncological expertise or experts to say, how is this likely to impact someone who's a female versus a male, or someone who's within this age range versus this age range? And those assumptions get fed into those sample size calculations, and they determine how wide or how big of a population you actually need. So the statistician really isn't determining like what the race factors or ethnic factors are. They're working with the key opinion leaders to determine what they are, and then to say, okay, based on that information, key opinion leader, we will likely need this many subjects to be able to say this drug works in 18 to 25-year-olds as well as it does in 65 plus year olds. Hopefully that makes a little bit of sense.
Yeah, it does. And it makes me think like, that we all have statistical analysis plans that go along with the protocol. So when you're thinking about that and you're thinking about, having these sort of subpopulation analysis done on these patients, is there, I know there's issues about multiplicity that are challenges, in the biostats field. So are you able to speak a little bit more about that? Like, how do you sort of minimize that risk when you're really looking at all these different populations and, applying different methods to that analysis?
Naomi, You got to be careful. You're going to be hired as a biostatistician here pretty quickly. This is pretty impressive. Like
It
that's
just, well, I'm so like, I'm fascinated by biostats. I would say I'm not good at math, so, but I have a lot of interest around it. So that's the reason for the question mainly. But
no, I wouldn't sell yourself short. I mean, you know what you're talking about. That was great. Absolutely. You're using all the right words like subgroup and multiplicity adjustments. Yes. So basically what statistics says is random chances everywhere. It's just a fundamental part of life. It's a fundamental law of physics. And so if I look at something once and then I look back at it again, maybe I just randomly happen to see a signal and maybe I make a false claim. Maybe I see, hey, the treatment's actually working on this subpopulation versus this subpopulation. So if you do this hundreds of thousands of times, you are going to see a significant result where one does not exist just by random chance. And so because this law of nature exists, we have to do something to account for this so that we're not just testing the drug on every little small population. And then we say, hey, look, it's working really good for this one small population, when really it isn't, because it was just a random chance. And that's called multiplicity adjustment. Again, without getting too technical, what you're able to do with statistics is to set a threshold, an alpha level is what it's called in statistics. And you can say, okay, if my p-value, if my statistical measure of significance is below that certain threshold, if it's small enough, then I know for a fact that random chance could not have come into the equation and that what I'm seeing is true. This treatment actually does impact these patients in this way. But the more you look, the more times you have to lower your threshold because you're continuously looking. You're just naturally going to see something. And so we have to be very careful. The biostatistician has to be very careful in terms of the number of times that we look, quote unquote, look at the primary endpoint and even the secondary endpoints to make statistical claims about the data we collect. So again, yet another evidence, another thing pointing to get your biostatisticians involved early so that they can adjust your sample size, they can adjust your analyses, they can adjust your multiplicity factors for however many times you need to look at the data throughout your clinical trial.
Awesome. That makes a lot of sense. Are you able to explain sort of, when you're in that at the end of a clinical trial, let's say it's a phase 3, and you have all this patient data, but you have patients that have protocol deviations versus those that actually complied with the protocol. How do you go about, like, what are the things that you look at, in order to, do you guys bucket these two differently? And basically, what's the approach there?
Yeah, it's a critical aspect to clinical trials. And it's a shame, but it's just the nature of running a clinical trial. So what you're talking about is as you go through the clinical trial and you're enrolling that sample, those handful of subjects, that small group of subjects into the trial, some of them, to no fault of anyone's, but some of them will follow the protocol perfectly. They'll take the drug when they're supposed to. They'll come back into the hospital to get measures taken when they're supposed to. Everything will go perfectly for them, and they'll be able, their data will be quote unquote clean. It will be meaningful, as in it followed the protocol well. For some of them, they will experience what's known as a protocol deviation. Maybe they'll forget to take the drug at a certain time, or maybe the site won't record a visit, and so that data is lost. And whenever that happens, you have to take a step back and say, not all patients within the clinical trial, not all subjects are the same. The data from a subject that did not have a protocol deviation, much cleaner, much more reliable, actually is telling the story as you needed it to be told in terms of, evaluating the drug correctly versus a subject that does have protocol deviations. The nice thing is that there's a way to account for this. Not all is lost. One thing you could theoretically do is to say, okay, you had a protocol deviation, you're out. I don't look at your data. And that's way too crude. That subject was still involved in the trial, that you can still gain something from them. So instead, what we do is we do these things called analysis populations, meaning that within the sample size, within the clinical trial subgroup, you have other smaller subgroups. And one of those subgroups is called the PPS, the per protocol set. And that is where you're only looking at those subjects that did not have a protocol deviation. And I should actually very quickly make a minor adjustment to this. And that choice of words is funny because protocol deviations are not all the same. Some protocol deviations are considered minor, where it probably doesn't have any impact on the primary endpoint, the primary objective of the study, at which point you can classify it as minor, and those subjects might still be included in the per protocol set. But there are other protocol deviations that are considered major, like they just didn't take the drug. That's a major protocol deviation. You clearly didn't follow the protocol at that point. And those subjects would be excluded from the per protocol set. It's the biostatistician's job to work with the medical monitor to determine, and other key stakeholders within the clinical trial, usually the project manager, medical monitor, and any other key members of the sponsor wants involved. It's up to the biostatistician to work with these people to say the protocol deviations, you've classified them as minor or major, who belongs into the per protocol set and who should be excluded from the per protocol set. So that way, when you run the analyses, you have analyses run on the per protocol sets, and then you have other data sets like the intent to treat population or the full analysis set that include some protocol deviations. So you're still able to draw insights from those subjects, but you're not necessarily using it to make a primary objective statement to test the primary null hypothesis.
Okay. Yeah, it's so interesting how, all these things that we have to take into account. It's sort of like you really need to pull in the key individuals and the key stakeholders from the beginning, right? Like when you're heading into thinking about what does a phase 1 look like and projecting long term, as to like phase 2, phase 3, and eventual FDA approval. It's sort of like you have to work backwards and include all those key individuals sort of from the beginning. Would you say
You're,
so?
oh yeah, you're spot on. So, and I probably did it a disservice earlier when I was talking about the phase 1 and the dose escalation and the involvement of the statistician. You're right. The statistician should still be involved there, not necessarily for the design of the dose escalation cohorts. Like they should be involved minimally there in terms of, do we want to do a traditional 3+3 or a Boeing design? But what they should be involved in is the full clinical program development plan. What is my target product profile that I want to be able to, what label claims do I want to be able to make at the end of my phase 3 pivotal studies? And in order to do that, you need to be able to know what type of pivotal study you're going to run. And in order to do that, you need to know what type of data you're going to get from your phase 2 study. And in order to do that, you need to know what type of data you're going to get from your phase 1 study. So you're absolutely right. You have to work backwards to design as early as possible the entire clinical program. And the biostatistician has a huge role to play in that journey of the clinical program.
Yeah, makes sense. I like to talk about technology and how, it's sort of revolutionizing the way we approach clinical research, clinical trials in general. Is there anything exciting happening with, I know there's AI that's sort of in the general public right now. It's sort of slow to come into clinical research for various reasons, of course. But is there anything sort of innovative that we're starting to see, whether it's from the biostats side or just other sort of angles in clinical research?
Yes. So very exciting times. I think not just for biostatistics, but for everybody in the life sciences industry, for the adoption of large language models, artificial intelligence, you name it. Data visualization is another big one. But even before I will speak about those in a moment, but even before I speak about that, there's actually a fundamental shift that's going on right now in the world of biostatistics, and specifically within the world of statistical programming. And those two functions are usually tied together. So when I say biostatistics, please know that I mean biostats and programming.
Okay, yeah.
And that fundamental shift is, up until this point, the dominant software, the dominant programming language for all clinical trial programming of data has been SAS. It's the type of transport files that the FDA accepts into their system for review of clinical trial data. And it's just the legacy architecture that a lot of large pharma, large device, and large CRO have built their data architecture around. And SAS is a great programming language. It's a great tool that does exactly what you need it to do for clinical trial programming. The issue with SAS is that it's closed, meaning that there really are the only improvements or the only additions that can be made to SAS are from the company SAS themselves. And they're doing so, don't get me wrong, like there are improvements to be made, but and they're doing it, but it's pretty standard with what you see is what you get. And this fundamental shift that we're seeing is really towards open source programming languages, specifically around R and Python. R being the main one, where we are seeing a lot of talk within the sphere, within the life sciences, biostats, and programming sphere of, hey, anything I can do in SAS, I can do in R, including generate more beautiful figures, more interactive graphs and charts that actually allow the reviewer to slice and dice the data however they might see fit. And so while I believe that SAS will continue to be the main language of choice within biostats and programming for the foreseeable future, R is starting to have its moment in the sun. And a lot of people, a lot of very smart people, are using this and generating very beautiful, very interactive graphs to help their sponsors and help regulators review their data more cleanly and clearly. So that's probably the biggest thing in technology on the biostats and programming front. In regards to AI and large language models, huge promise and really an exciting time. And a lot, of course, the very first question is, what does this do for jobs, right? Can I have AI just program all of my data sets and program all of my tables, listings, and figures? I want to answer that one immediately and say, not right now, and probably not for a long time. And I don't want to make any predictions about that, because who knows, maybe it's an exponential curve. But what we are seeing specifically within ProPharma and in my own experience is it's not here to replace anybody's job. It's here to augment it. And so if ever I have a very sticky piece of code that just for whatever reason I can't write, I don't know how to do it, I can go to Copilot or I can go to ChatGPT and say, hey, I have to transpose this data set. These are my variables. Can you tell me how to do it? And it will give me code that, while isn't perfect, is at least a good starting point for me to then say, okay, now I know that it's proc transpose within SAS, for example, and I can start programming around that snippet of code. So AI and large language models absolutely have their place in the life sciences sector, and specifically for biostats and programming. It's a great tool to get you off the ground when it comes to, hey, what statistical test should I use? Or, hey, help me write this piece of code so I can generate this survival curve. And we're able to leverage that pretty successfully so far, I think. So that's kind of the biggest advance of technology currently going on.
Yeah, that's really exciting. Your first point where you're talking about SAS versus R, you said it was open source. Does that mean there could be issues with like data privacy or anything like that when you're starting to code into something that's like, oh, anyone really has access to this type of programming? Is that any sort of
Yeah.
concern?
No. It's very good question. But when I say open source, I mean just that the functions within R, anybody can contribute a new package. Anybody can write a new little script that says, hey, this package of R will take your data and do X, Y, and Z to it. And so instead of having to write that code yourself, you just call this new script, this new package, and it will do those things for you. The biggest example of this is the R Shiny package, which enables for very interactive user interfaces. Now, instead of you having to manually code that yourself, you can just call in the R Shiny package, and there's a handful of parameters you have to provide. So open source really just means the code that is behind in terms of the functions that the programming language does, not necessarily the actual clinical trial data. I should really highly note this, that every CRO company, every pharma company, every biotech that houses clinical trial data, we have to abide by CFR compliance, specifically 21 Part 11. And that means that all data has to be secure, good clinical trial practice validated, GXP validation. And you have to really show that it's auditable, traceable, and legible. So you actually have real security parameters in place to protect clinical trial data. So regardless of if you use an open source programming language or not, if you're doing business with a clinical trial, with clinical trial data, the regulators absolutely require you to have that buttoned down behind a firewall, super secure, just to kind of put everybody's minds at ease about that.
Yeah, no, definitely. And you bring up a good point. I mean, there's a lot of things within the clinical research industry that, is we have a regulatory oversight of the FDA on. And so what other, you brought up a good one. What other sort of key points that do biostatisticians have to really make note of to make sure we're in compliance with the FDA?
There's a lot. Probably the number one would be the data requirements, the conformance guides, to make sure that the data that you have collected is organized in such a way and stored in such a way and presented in such a way that facilitates their review. Probably the most well-known one is the CDISC standard. So if you are submitting data to either CDER or CBER, those divisions within the FDA. CDRH currently does not require this as a data standard. But if you're submitting to CDER or CBER, you have to submit your clinical trial data in what's known as CDISC format or SDTM and ADAM format. And basically what these things are data structures that allow the FDA to quickly review clinical trial data regardless of the clinical trial. So what happened when, I forget when this started, back in the early 90s, when the FDA opened up to electronic submissions, when they started receiving trial submissions in electronic format, they kind of threw their hands up and were like, oh my goodness, this is way too much data. Like, the number of submissions skyrocketed because of how easy it is to just submit electronic data as opposed to submitting reams of paper. They kind of threw their hands up and said, we need to standardize this. There's no way we can look at everybody's data in a timely fashion unless it's coming to us in a standardized way. So an independent group was formed, CDISC, and they provide guidelines for how data should be structured and how it should be organized. And so the SDTM format is really the human readable format. It enables the FDA readers or reviewers to say, hey, I know where demographic data is. It's in the DM data set. Or, hey, I know where laboratory values are. It's in the LB data set. And then even further, within each data set, you have specific variable names that are common throughout all data sets, throughout all trials. So it really facilitates a very nice, very quick review of the data. And then the other one is ADAM. So once you have the SDTM data sets, you also have to have ADAM data sets, which is the analysis data model. So it turns out when you organize data that's good for a human to read in SDTM format, it's not so great for a computer to read. So you want to take that SDTM data set and transform it slightly into what's known as the analysis data model. And then you can feed those data sets into the computer to say, hey, run me this linear regression. Hey, run me this mixed model of repeated measures. And then it generates the p-values from that model that you asked it to run. So those, I think, out of all of the standards and all of the requirements that the biostatisticians have to play by and abide by, knowing the data format standards is critical because it's a majority of what we do is organizing, analyzing, and reporting the data. So you have to know the data standards.
Yeah, definitely. So, well, how do you stay up to date with sort of like the latest or the best practices, latest developments in your space?
Yeah, no, it's a challenge. It's for sure a challenge because there's so many news outlets, there's so many avenues to receive information and to parse through it. So fortunately, within ProPharma, we have a division that is dedicated to receiving any update that the FDA releases. And the FDA does a great job of releasing via email, like, hey, these are the new guidances we released. These are the new regulations that you need to abide by. Or here's a regulation that's in draft and we're accepting comments. They do allow people to comment on their guidances. So this division within ProPharma helps us to stay on top of these came out. This is probably impactful to medical writing and probably impactful to biostatistics. We then have within our division, we then have team members who go into those reports and say, yep, this was flagged correctly. This does impact biostats. And here's the SOPs that we either need to update or add a work instruction to ensure that we are abiding by the new guidance and the new regulations. But it's a full-time job. I mean, you really have to have a division dedicated to making sure you're staying up to date because it's a highly regulated field. And the worst thing that can happen is to run a clinical trial, submit it, and the FDA reject it because you didn't follow what they required for that type of study.
Yeah, definitely. So it's likely that a lot of sort of companies have their own internal team. Have you heard of any sort of, and I'm asking because I haven't seen it, but maybe you have any sort of just general platform for clinical research professionals of any kind to go in and get sort of updates? You said there's some news outlets, but it seems like everything's sort of like sparsed out in terms of information like this.
Yeah. When I say news outlets, I mean, there's a couple of websites that come to mind, but none that really cover like FDA guidance. It's more of those websites
Mm.
cover like new drug applications and who received funding, things like that. So in terms of, stay up to date on guidances, the best sources are certainly the FDA themselves. And you can actually go on their website. I don't know which section of their website by heart, but you can go in and say, hey, here's my email address. Email me anytime you guys have new guidances that are applicable to these types of drugs, and you can select what you're interested in. Like, I work in the cardiovascular space, for example, so I want to know about cardiovascular, or I'm in the oncology space, so I want to know about any guidelines that impact oncology trials. So I think the FDA is probably, for any individual who's not part of a larger organization that has these divisions that are geared towards staying atop regulations, my recommendation would be go to the FDA website yourself and throw your email in there and ask for them to send you emails
Mm.
weekly about any new regulations that come out.
Yeah, that's good advice. So now switching gears into like the career of biostats and someone who's interested in it. So what do you think would be some sort of key skills to have to kick off a career in biostats?
I think that the most important thing would be make sure you derive joy from puzzle solving. Every day is a new puzzle. Every day is a new trial trying to do something new in a population that is either underserved or hasn't been served before. And so you have to really use logical thinking to say, okay, this is the therapeutic area. This is what the sponsor is trying to do with their label claims or their target product profile. Let me go hunt for what the FDA needs to see. And now let me put those puzzle pieces together to determine the best way to analyze this, and then let me execute. So if you find joy, if you truly like solving puzzles with math and have a mind for it, it's a phenomenal career. I really am very lucky because what my team and I are able to do on a daily basis is to improve the health and safety of patients. What an incredible use of our time and our talents to
Mhm.
support the world. It is challenging. I absolutely want to make that very clear that there are days that are difficult. There are days that are easy, but on the average, I think that it is a challenging career. But if you have the passion and the joy for solving puzzles and helping people, and you have the mind for mathematics, the next best skill set to have, the next best thing, believe it or not, would be communication. Because the clinical trial is, I'll say it again, it's such a complicated song and dance. Hundreds of people behind the scenes work on a phase 3 trial, hundreds of people. And so being able to know when to raise your hands and say, hey, I don't have this from data management. I really need to understand what the critical variables are and how they're being collected. Data management, can you please provide that to me? Knowing how to ask for that, when to ask for that, and having the confidence in yourself to be able to do that is a critical skill set to have in order to really grow your career as a biostatistician and climb the ladder, so to speak, to be able to help guide other statisticians and use your skills for an even wider range of services.
Yeah. Yeah, I love it. I think a lot of those skills is really, not just for biostats, but other
Yeah,
positions within clinical research. We're
totally
always,
agree.
even if you're talking about a monitor, you also have to sort of problem solve, figure out, what's happening here, kind of figure out the root cause, right? So, in talking about sort of like mentorship, have you had anyone in your life during your career trajectory that has really been of influence to you?
Yes, I have. I've been incredibly fortunate. At every place I have worked, I have had at least one mentor or person where I have picked up just an incredible amount of tips, tricks, skills from. And my first job at that really small startup CRO, it was the owner of it. I learned the value of hard work from her. She really taught me that what we're doing is important and we are helping more people than we could ever even possibly imagine. And so we have to give this our best effort to ensure that those people are treated well and that the FDA is given the full story to make their decision. Because maybe the drug truly doesn't work. And at that point, we don't want the FDA to approve that drug. It's up to us to be able to say, this is the best possible package we could have created of the data that was collected about this drug. It's now up to you, FDA, to make the decision. Come to us and ask us any questions you have about what we did, why we did it. But it ultimately does rest on the FDA, and we have to do our part to help them make the best decision possible. Then from the sponsor side, when I was at the sponsor company, my immediate supervisor really taught me what it was like to manage people. That was my first foray into management. And he taught me all about how everybody is different. And that's a very good thing. You don't want to treat everybody the same way. You cannot do that. You have to listen to people and understand how they like to be led and how they like to work. And then you have to give each of them their own space and their own ability, and to trust them, to know that they can do it, and to really instill trust in the team. So along the way, I've had a tremendous amount of mentors that I am so, so grateful for. And they have not all been biostatisticians. At my current company at Proforma, my mentor he constantly challenges me and teaches me to think very broadly, but at the same time, think very specifically. So keep the general in mind, but focus on the direct problem at hand and how that would impact the general. So you always are thinking big picture. That is, so he's not a biostatistician, but he is someone who I have learned a lot from over my time at Proforma currently. Yeah, huge believer in mentorship. If you don't have a mentor, highly recommend you find someone that cares about your growth. That's important in a good mentor. They have to care about your growth. And they have to be someone that is where you want to be, right? They have to have traversed the course that you plan to traverse, or at least not necessarily follow the same path, but at least get to the level that you want to get to. It's helpful if your mentor is already there.
Right. I think, there's big companies that have mentorship programs, but the way that it's set up, sometimes it's you get paired up with someone who may be, a VP or senior director, but they may be in a completely different scope, or like they may be in marketing, HR, like whatever it is.
Yes.
And they do it on purpose, I know, because it's like, well, you want to be as transparent as possible to this person and have it not be like someone you interact with generally in your job scope. I would say, it is difficult sometimes to get mentors. Do you suggest maybe using platforms like LinkedIn to be able to sort of connect with individuals who might be interested in mentoring you?
Yes. And your point of there are mentorship programs out there where they do line you up with a mentor who's not necessarily in your vertical. And I think there's real merit in that. I think there is real value there. You get to see a broader picture. Going back to our initial discussion about, if you're starting your career, you want to get as wide of a view as possible. So that way you can determine where you want to go deep. And if you're at that point in your career where you don't really know where you want to start, having a mentor outside of your current vertical, that's a great way to go. I think that absolutely makes a lot of sense. In terms of securing a mentor or finding a mentor, if your company doesn't provide them for you, LinkedIn is the tool, like, as far as I'm concerned. There's a lot of people out there who are providing real material, real helpful items, typically for free. Reach out to them. Say, hey, I'm interested in doing this. Would you recommend that? And do you have 15 minutes to share with me? And then if you guys hit it off, maybe you found your mentor. But I think LinkedIn is the tool to find someone who can guide you and help you as you get started or grow your career.
Awesome. Well, sort of the last question, what advice would you provide someone who's aspiring biostatistician?
Oh man, I should have prepped for this one. This is a good one. What advice would I give? Outside of what we spoke about already in terms of going wide first, because within biostatistics, there are a lot of different nuances. If you work on the sponsor side, for example, you are very likely to be doing more study design work and more study reporting work. So you're doing the bookends, whereas if you work at a CRO, you're much more likely to be doing the actual conduct of the study. You're much more likely to be creating those CDISC data sets and those TLFs that ultimately go into the report. So the advice there is know which one you like better. And if you don't know, then just randomly choose. I would recommend a CRO to start, because if you go to a CRO, you are going to work for a multitude of different sponsors. And that gives you access to a multitude of different therapeutic areas, theoretically, depending upon how your CRO is structured. Some CROs are structured by therapeutic area, so you might just get assigned one therapeutic area. But suffice it to say, the advice is, if you're starting your career, try everything if you can. Try and see if you can't design a study. Try and see if you can't conduct a study. Try and see if you can't report a study. Try and see if you can't work within oncology, cardio, gastro, any of the therapeutic areas. See as much as you can, and then dive deep into one specific area of focus. In terms of advice for developing skill sets, actually, no, I know what it is. It is to read ICH E9. This is the International Council of Harmonization. This is the guidance that the FDA follows that says these are how you do statistical elements within a clinical trial. It will teach you about what the expectation is for sample size. It will teach you about what the expectation is for null hypothesis testing. ICH E9 is the document that we require within our division at ProPharma. Every new statistician reads, and they have to present to us on it. They have to tell us, hey, this is what I'm thinking. This is what I think this means. Do you agree? And we have a discussion, because it's that fundamental. So career advice is try and see as wide as you can. And then once you land the career, read ICH E9. Make sure you're familiar with it. Make sure you can recite it in your sleep, because it's going to guide everything you do.
Awesome. All right. Well, thank you so much, Robert, for joining me today. I loved our discussion. If people are interested in connecting with you, where can they find you?
Yeah, great question. LinkedIn for sure, Robert Rashford. I also have a website, like I mentioned earlier, betterbiostats.com. Free resources currently for biostatisticians and statistical programmers. But I also have a form on there where you can message me directly and say, hey, do you have anything about anything, X, Y, and Z? And I'll usually respond to you within 24 hours. My sole goal, my sole purpose in life is to help others achieve their best and really help the life sciences industry improve the health and safety of patients. So find me on LinkedIn, find me on betterbiostats.com.
Hope you enjoyed my conversation today. If you're interested in these types of discussions, please subscribe to our YouTube channel. We also have these posted on Apple and Spotify podcasts. For more insights and updates, you can also sign up for our newsletter at TrialTalksInc.com.